Thanks for your patience. Thank you for the introduction, Mr. Voss. Yes, I'm from the University of Sherbrooke. I will talk about the bacteria. And here we go. As you're all aware, despite being a model organism for more than 30 years, there is still a lot of unknown in E. coli's genome. This is important because the so-called white gene hide functions and interactions hindering our capability to study the function of genes and pinpoint eventually critical vulnerabilities. The classical way to study the function of a gene is to delete it and assess its viability. Yes, I'm one behind. The viability of a resulting cell is to assess the viability of a deleted cell in different conditions. This strategy was used to produce a KO collection encompassing more than 3,800 single gene deletion strains. However, when the deleted gene is in genetic interaction with another one, which is the case, for instance, in redundant pathways, as it is happening a lot in the complex E. coli genome, its function will likely stay uncovered. We then decided to go from single gene to genome scale interaction map to elucidate more gene functions by using the HTTM method that I'll explain in a few slides. Because HTTM is based on transposon insertion sequencing, or TIS, let's first revise this experiment to make sure everybody is on the same page. So TIS, it starts with a transposition vector transformed into receiver cells, here represented by a circular genome. The single-use vector is only expressed once, leading to a single transposition event in the cell at a random position of its genome. The transposon, if landing in a gene, will interrupt transcription and lead to a knockout or insertional inactivation. Each cell, again represented by its circular genome, contains a single transposon insertion and is then grown on a rich medium, leading to the disappearance of the cells containing an insertion that disabled an important gene. Note that even a moderate fitness disadvantage would cause fading, since each mutant competes against one another. For instance, here the insertion in the green gene caused the death of these two mutants, leading us to think that it's an essential gene. To identify which genes are essential, we sequence the entire population from the insertion point to the surrounding genome region, producing an insertion map at a given position in every cell, represented here as linear genome, that are then aggregated into population-wide tracks, showing the distribution of transposon insertions. As you can see, this track shows us where an insertion can be found and where the interruption never occurs, representing respectively non-essential and essential genes. But how does this help us identifying relation between genes? We will get to it soon, but first of all, I'll quickly define what I call an interaction. Today, I will focus on negative interactions involving a loss of fitness of the double mutant, which are also called synthetic lethality. As an example, let's consider this classic interaction involving the acquisition of the glutamine, an essential metabolite. It can be acquired through either transport or biosynthesis, and the cell doesn't mind which method it uses, as long as there's enough of the so-called metabolite available. So the acquisition methods are functionally redundant. Both components, transport and biosynthesis, can be deleted individually without affecting the cell's viability. However, knocking out both components simultaneously leads to non-viability. In a TIS experiment, this would be reflected in the data like this. Gene number 4 becomes essential in a strain lacking one of the two components. So far, I've described the classic TIS methodology, but however, we wanted to apply this approach to every single mutant in the KO collection, combining single gene deletion strains with insertional inactivation. This was not feasible with the standard TIS approach, so we had to rework it into an upscale version. My colleagues in the wet lab adapt this method to be suitable for plate processing and optimized the library preparation, while I developed the streamlined pipeline for handling these sequencing data. This upscale version of TIS is what we now call HTTM. To summarize, HTTM is a way to improve throughput of traditional TIS, allowing us to deploy it seamlessly in large collections such as the KO collection. So once a CTM experiment is completed, we obtain a track of insertion positions for each of the KO single deletion strains. Sorry. The resulting data would ideally look like this, with extremely clear patterns, but in reality, it's more like this. It's a bit noisy. The coverage varies slightly, but while this is a minor drawback of high throughput, it is largely compensated by the large number of samples we have. We then count the number of insertions for each gene and strains and place the count into a matrix like this. So where the diagonal is null, since the deleted gene cannot have an insertion count, and note that the column and row were swapped. Since there are approximately 4,000 genes in E. coli and 4,000 strains in the KO collection, we are probing 16 million of digenic combinations, looking for relatively rare genetic interaction events based on similar studies in the yeast. So when we zoom on a region, we can see the insertion value for the given gene G, so in the row, which is distributed this way, where the vast majority of the normalized counts are low, and the gene deleted in the KO strains associated with extreme negative values for a given gene, for instance, delta S here, are declared to be in synthetic lethality with the gene. Now, remembering the example we discussed earlier, if the transporter components on the left side are deleted, GLNA become essential. And here, the arrow represents the direction of the relationship. On the right side, deleting GLNA makes all three transporter components essential. This interaction network should look something like this, where nodes are genes and edges represent interactions. So after processing the entire matrix, we obtain a large network of approximately 47,000 interactions with a p-value under 1%, involving 3,602 genes, or more than the 3/4 of the genome. Here, the center of the network represents a more dense area, highly connected genes, while the outer rim consists of genes that are less connected. So now, how can this network help us elucidate gene functions? I have two insights on this. Among these relationships, we find well-established interactions, such as between GLNA and GLN-HPQ, exactly as expected. And shown here is the track of the raw insertion data for several deletion strains. So GLN-A, GLN-P, and H are there, while GLN-Q was not available at the time of I made this figure. Each row is a mutant. The x-axis is genomic position. You can see clear gaps region with no insertion in precisely those genes we later identify as having negative interaction. It is worth noting that we also began acquiring these same data in minimal mediums by opposition to this rich medium I'm presenting now. However, we did not observe the GLNA interaction network, which is expected since there is no glutamine in the medium. So the transporter is useless and cannot be in interaction with the GLNA biosynthesis pathway. But more intriguingly, we observe new links between GLNA and both GLN-D and G, genes that are also involved in the glutamine pathway. This suggests that we may either have greater sensibility than previous studies, or that the growth condition we use have led to new interactions. So the glutamine acquisition network is also significantly connected to other related genes, highlighting the high functional redundancy in E. coli. But more interestingly, it is also connected to numerous poorly characterized genes. The direct impact of their deletion on the essentiality of different components suggests these genes play a role around the glutamine acquisition pathway. Therefore, I'm investigating how to propagate these functions to Y genes, as we have validated hundreds of known interactions, leading to thousands of relations involving yet to be documented Y genes. So we also observe a large number of highly connected genes. On average, each gene has 26 interactions, regardless of the direction, but many genes exceed this. So in this heat map, The blue nodes represent genes with above average connectivity, and the red nodes indicate below average connectivity. On the x-axis, nodes with inward arrows represent genes that are affected by others. And on the y-axis, nodes with outward arrows represent genes that affect others. So I'd like to draw your attention to the 513 genes with above average outward connections and below average inward connection. This indicates that many genes are unilaterally responsible for causing essentiality. Based on this, I hypothesize that genes with more outward connections are more likely to be higher up the regulatory hierarchy. So to test this, I extract the key regulator CPXA, a well-known high-order regulator involved in membrane stress sensing. I then traced 5 outwardly connected genes from him, and then the downstream targets over several rounds. We consistently observed a progressive decrease in the outward connection amount as we move down the regulatory tree. This suggests that CPXA not only regulates many genes, but those genes in turn regulate others, amplifying the effect within the genome. I observed similar pattern when for many other regulatory genes, such as this list, which is expected. However, things become intriguing when the root of this regulatory tree is a gene coding for a non-characterized protein like YMIA. What does this tell us about its role? Does YMIA share regulatory properties similar to known genes, or can we infer general regulatory functions from the topography of this network? Unfortunately, I don't have concrete answers yet. These questions are still in the early stages, and the data are still fresh out of the proverbial oven. So that said, we are confident that these interaction hubs are critical therapeutic targets since they seem to have an important impact on the regulatory equilibrium. Of course, at some point, it would help to know the function of these crossroads so we can target their mechanism properly. So I'll admit, I was a bit ambitious when I suggested this title for this talk back in the spring. While we uncovered some promising genetic interactions, we are still away from fully eliminating the entire regulatory landscape. And there is so much more to explore. So this is also why we're currently producing similar datasets for other growth conditions, such as minimal medium, as I mentioned earlier. And we are currently translating the method to other organisms that also have a single gene deletion collection like the KO. But before this, I am still in the search of creative but concrete ways to analyze these rich data in silico. In this vein, I'm very excited to start investigating these interactions across pangenomes within the frame of a collaboration with Dr. Fiona Whelan's team in Manchester in the upcoming months. We will try to determine if genes involved in relations are also subject to co-occurrence among different strains, species, and eventually clinically important bacteria. For now, I welcome all your insights and ideas. Please feel free to reach out. I'm open to suggestions and further discussion. Finally, I would like to sincerely thank all the funding agency for their support, as well as all team members of both my co-director, Sébastien Rodrigue, and my main lab under Pierre Saint-Jacques. Thank you for your time and attention. I look forward to continue the conversation. Cheers.
So that was a really interesting data set.
Thank you.
I wonder if you could talk a little bit about how surprising it is to see so many synthetic lethals. It seems like a lot.
Yes.
And we should talk after about something else that I've seen with similar. So.
Nice, of course. Yes, thank you for the question. First of all, I mentioned briefly that even a small fitness cost in this context will fade the mutant very quickly. So we're doing a couple of passages. As soon as a cell loses the capability to compete against the other mutant, it will disappear. So maybe we are seeing very slightly deleterious effects. Also, there is a huge computational process in this, so we are using statistical thresholds that are not quite perfect as we talk. So we can have like for until maybe 100,000 relation as well. So yeah, those relations will be tested in vitro eventually, of course.
Following up on that question, is it a possibility that some of these knockouts are, so you're looking at lethal, non-lethal only? Or, yeah, so could it be that some of these knockouts are false positives, meaning the interaction is not a direct one, and therefore you're identifying the direct one as an indirect, eventually that might cloud the way you map your system? So how do you tease that out?
This is a very good question. We are beginning to handle these networks, and we're trying to figure out how to handle them. What you're saying is that two nodes that are interconnected with another node between them.
At least one,
yeah. What does it mean? I'm not sure at this point. We know that the interaction can be of any nature. In fact, we are not expecting protein to bind together or something. It can be very diffuse and distal relations. So does a gene have a relation with children of farther than one leaf away in this network? I'm not sure yet. So thanks.
Any further questions? Okay, in that case, well, thanks.
Thank you very much.